Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. And now here is part 2 of our conversation with Dr. Andy Vick.
So, Andy, what has been one of maybe the biggest surprises to you in the industry across your career?
Oh my, you're taking me on a 26-year journey. The biggest surprises. I don't know that I've had any major surprises. That's probably because every day I'm at least a little bit surprised, and so it's normal for anybody else. Yeah, I don't know that I have any massive surprises. I do think, there's 1,000 kind of ahas that happen in 26 years. I mean, I'm old enough to have lived through De Janeiro, and I remember when the day that hit the headline, I was quite a bit surprised because and boy, I'm not that old, but I'm old enough to know that I particularly love the biologics development, and part of that is because there still is, and there certainly was, even at the beginning of my career, very much a pioneering, kind of blazing a trail westward feel about it, that we were just learning so much. And I think De Janeiro happened fairly early on in that kind of that evolution. And obviously, as it related to immunomodulators, we learned a lot, and particularly predicting what would be a safe initial dose and a first in human clinical trial. So I remember being surprised by that more in a humbling way, which that's not new for me. Just realizing, I think that there's a number of times in our scientific careers where we're reminded you just really don't know as much as you think. Humble yourself and start listening and learning more. I think even if I was to transgress a little bit about that too, I think we're even in my career, the surprises, which has not become too much of a surprise. It seems like technology also is like fashion, and it comes in and out of fashion, every 10 years or so. So I'm seeing some of the cyclicity of that already. And even now, I would say not a surprise, but it may be a pleasant resurfacing as pharmacognosy seems to be quite hot again, and the study of natural products as therapeutics. And I'm a big advocate of that because I also think sometimes probably there's a lot more solutions. There's a lot of very potent therapeutics that are right out there in this wonderful ecosystem that we have called Mother Nature. And I think sometimes it's our desire as people to always to be moving up, to try to make it better. And it's pretty hard in my experience to really try to improve on Mother Nature. So I am enjoying maybe the aha of the kind of the refocus on natural products and what we can learn from botanicals and venoms and various other products that we have in Mother Nature that could be very profound therapeutic products. I mean, that was even maybe one of my, but certainly an enjoyable, because I'm a bit of a reptile nut. We may come back to that, but certainly a reptile nut. And so being able to be a contributing scientist to the first generation of Byetta, exenatide, which I think, as you all know, was exenatide was a high homology to human glucagon-like peptide 1 that was identified in the saliva of a Gila monster, one of 2 venomous lizards in North America. So I think that's a great example of, I think folks often forget that as a natural product that has come to be one of the most potent type 2 diabetic products. And we even leveraged some of that learning in one of the products that I was an inventor on, which is Trulicity or dulaglutide, which is the long-acting, an FC fusion of a modified GLP-1. Working with a company now as a consultant that their whole portfolio is venoms. I mean, if you can think of something that has fangs or a needle or teeth or anything that could deliver nematocysts, anything that could deliver venom, they're looking at some of those products, and a lot of those are ion channel inhibitors. So maybe one other aha. Boy, there's a lot. I think we're yeah. Even I got 10 or 12 that are coming to my mind, but there's so many. I'll probably just pause there. So you kind of get a sense, nothing that really well, maybe one other one. And that's just which is a real blessing too, because even at the beginning of my industry journey, I think anytime you heard the C word, cancer, you kind of just assumed that it was going to be a death sentence. We just, it's kind of fitting. We just unfortunately talked about glioblastoma, so we clearly have some more work to do. But but also when you just sit back and you think of how far we've come, so that in many instances, these cancers are very well managed, especially if they're identified early with the tools that we also have to diagnose early. And so again, there's much less of the fear effect that cancer has, circa 2024 than it did circa 1980s and 90s. Same thing comes to mind when you think HIV. I mean, that was my kind of generation early in my industry career, and so just just how impactful some of the products have had over the past quarter of a century, and that's not insignificant, because as we all appreciate on the phone and hopefully in the ether, is that this drug development process isn't overnight. I mean, it's probably a good 10 years from the glimmer in a biochemist's eyes to on the market. And so knowing that, and then kind of sitting back and appreciating the scientific advancement that we've had in the past 25 years, I guess maybe that's my not so short answer.
Yeah.
That's probably the biggest wow for me, and just thinking what's around the next corner, which I get really excited even about. We've kind of been chasing the tail of Alzheimer's disease and a number of other neurodegenerative diseases, but I just feel like, we're coming into some, we're seeing some real advancements there. So 5 years, 10 years from now, maybe we'll also be sitting back and thinking how far we've come in some of these disease states where we knew so little. And that also, again, that circles back to just being humble, being, having a unquenchable thirst to learn and to challenge and to innovate, and not just be satisfied with good enough. But we're in the business of developing great, and there's a logarithmic difference between great and good. So
I kind of love that last example there, Andy. Actually, I do love it, because thinking about in the late '90s when you and I got into this business, it was all about the creativity was what new shape of small molecule can you come up
right,
with, right? And
and
then you
yeah,
do
the nth in class, right?
Yeah, and then you who could copy it the best, and and then when the markets, but now with the small molecules are every bit as important, but we're using them in interesting ways like protein degraders. We have obviously biologics. We have multi-domain specificity, gene therapy, cell therapy. You guys are delivering not just a payload like a traditional ADC, but you're delivering or carrying to targeting that peptide to bring a macrophage in. And it's just like, it's imagination. You just use your imagination on a way to tackle a disease and then find a way. So
well, one
it's
thing
amazing.
too,
We're so lucky.
that absolutely, and even if you look at the last, I'll say 5 years of FDA approved drugs, it's something I routinely do, when giving different College of Pharmacy updates. But just the portfolio changes. Not only is it shifted from a very small molecule centric to a more biologics in the portfolio, but if you even look at the last 5 years and you look at the percentage of first in class, breakthrough therapeutics, rare and orphan disease, then again, my memory on these is not going to be exact, but they're in the 30-35% of the approved drugs each year, which 10 years ago would have been a fraction of that. So I'm really pleased to see some of those statistics and the direction that they're going for our industry. So.
Mm-hmm. Mm-hmm.
Excellent. And you mentioned something that was on the list of questions I wanted to touch on, talking about venoms. A couple of months ago, you visited Ashland University to discuss exactly that: snakes, venoms, pharmaceutical science with future toxicologists. Tell us a little bit more about that experience and what you did there.
Yeah, so I have. I guess I'll start with the hobby side. We'll just get the creepy out of the creepy stuff out of the way. But yeah, I've been almost a reptile enthusiast or hobbyist, whatever the right word is, for almost 40 years, and so have, for reasons that I don't even fully know even today, but I have just a passion for reptiles, and have accumulated a small and pretty rare collection of reptiles from around the world. A lot of old world rat snakes like Thai bamboo rat snakes, mandarin rat snakes, rhino rat snakes, but then a number of other species as well, and try to do a little captive breeding on those, not only from an education standpoint, but also just to do, my small part to maybe minimize some of the pressure on the wild populations of these specimens, and and um, and just try to do a little bit of education. I mean, I think that one thing I found in 40 years of this, kids are rarely afraid of reptiles. Their parents are. And so then there is a learned fear of reptiles, but young kids are fairly fearless when it comes to reptiles. So fear unfortunately is something that's taught. Uh, and then also just kind of reptile husbandry and medicine. If you're going to, and so again, if you have the privilege of keeping an exotic species, just making sure you keep it not only just adequately, but you meet or exceed its even environmental husbandry. And I'm a little bit surprised too that even large vet schools, I'll even pick on one in my state. In 4 years, a vet student may only get an hour or two of reptile medicine. So there's just not much known about kind of reptile medicine. So I've amassed a bit of it, just trial and error over 40 years. And so anytime I have an opportunity to give back, and I'm also passionate about helping young professionals navigate their career, and however I can bless them. But anyway, I can kind of combine, a passion for the pharmaceutical industry, a passion for young professionals, and a passion for reptiles. That's like winning the lottery, a trifecta. And that really was the Ashland University. They have a toxicology club. And it was kind of a multi-focused hour and a half. Some of it was just career development, professional development, but also give them a scientific presentation. And one of the young students in the group knew I was a reptile nut, and so she's like, "If you could tell us about the pharmaceutical potential of venom, the pain and protection of venom, that would be maybe a good way to just kind of warm up the group." And took a few reptile friends. So yeah, that was just maybe a couple of months ago, and have done that at a number of other universities. Even did that at AAPS, which I think I was maybe the pilot experiment for their 5-minute Ignite series, I think is what they call it. And 5 minutes, as you can tell, for a guy like me, is like an answer to one question. So 20 slides, they auto-advanced at their own speed. I kind of purposefully didn't prepare, which was a big mistake. But the one thing I remember loving is most of AAPS staff hated snakes. Sharon Pashon, who's retired, but a wonderful woman, hated snakes more than anybody. So after I showed a couple of very gory pictures of some of the pain of snake bite and envenomation, like necrotizing fascia, just skin and muscle melding off your bone, I happened to have a box full of rubber snakes. And after I had gotten the audience nice and gored, grossed out, I stepped towards the audience, pretended that I fell, and dumped the contents of this box and all of the plastic reptiles into the crowd, which created a pretty lively reaction, but one that we still talk about years thereafter as maybe one of the best Ignite sessions. It definitely had an element of a fear factor, but again, I digress completely. So,
Andy, you don't have one of your reptilian friends close by, do you?
I do not. Actually, we're in a room that is sometimes my quarantine room, and so I do have a cage over here otherwise, but like even a young young girl who had visited the house to see the collection drew a picture of her favorite snake. I have pictures of snakes even around the room, but I'm afraid if I move the camera, I'll never be able to move it back. But a future meeting, I can bring some animals with me.
Can't wait.
There you go.
Can't wait.
That'd be great. So, you're an adjunct professor at Ohio State. Are you teaching? Is there a regular class you do? Tell us about what you're doing there.
Yeah, that's another part of the not liking to be bored. So I figured I had tried contract research. I had tried the innovator side of the industry. The only thing I needed to kind of sink my teeth into academia. So giving back to the Ohio State University that had given so much to me was an easy choice. And so I think maybe for about 5 years now, being adjunct in the College of Pharmacy, Division of Pharmaceutical Chemistry. And that the scope of that role is pretty flexible. So it can range in giving kind of guest lectures from time to time. Sometimes those are scientific in nature. Sometimes they're just career and professional development for their students. That could be P1s in PharmD or their graduate students. That could it could be scientific topics as well. It also included kind of serving on their Dean's Corporate Council, which is a little bit like the board for the College of Pharmacy with outside influences from retail pharmacy, from the pharmaceutical industry, and other disciplines of this world of pharmacy. And then also, as you can imagine, academia is becoming an increasingly kind of a collaborative partner for industry and academia. So academia being kind of the true innovators, and pharmaceutical being the developers. So as assets come in from time to time, largely those that have been developed in academia, sometimes I'm a part of a group that goes in there and kind of evaluates those from a diligence standpoint, helps them particularly develop maybe an ADME tox strategy to develop that product from lead optimization or candidate selection into the clinic, and a number of other things as well that probably escaping me. So as you can imagine, it's flexible, and wherever I can be of benefit to them, I try to be. So
awesome. I have an Ohio State story for you real quick. Was just at a conference a couple of weeks ago in Columbus, actually, for MedCon that they do every year. And there's a lot of FDA there, and they're talking about innovations and regulatory and all sorts of fun stuff. Well, they had a couple of patient stories, and one was a young lady she's just about to graduate this year from Ohio State. And when she was a baby, she had open heart surgery and had problems with one of her heart valves. And the doctor said, someday you'll need to have this repaired again as an adult. Don't know when. You'll know when it happens. And so lived her life, had everything normal, was into athletics, played a ton of volleyball. I was able to get a scholarship to Ohio State to play volleyball freshman year. Super psyched, living the dream, that she wanted to do and play for the Buckeyes. And the first round of heavy conditioning that she did, she fell shortness of breath, and she just thought she was out of shape compared to her peers. Mm-hmm. And so she talked to the trainer and then went to the cardiologist, found out that was the signal. It's like, crap, now I got to go back. And she was devastated because she had to go and have open heart surgery was the round of treatment. Well, she came across a clinical study by Edwards Life Sciences that were basically putting in an artificial heart valve through a catheter, which sounds crazy to me that you can do that successfully, but they did, and they were in phase 3 trial. Talked to her about it, and so she was very excited about this opportunity. Her parents were not so much. They're like, no, surgery safer, but she was 18.
Yeah.
So she had the opportunity to kind of decide on her own. And though she's a little scared about it, she really wanted to do the clinical trial. Long story short, she did the clinical trial, had a successful replacement of her heart valve via catheter in the trial, and was able to, rather than have this 6-month-long recovery and miss the whole season, was able to play in her first game as a freshman with a new heart valve.
Just weeks later, probably. Couple weeks.
Basically 2 months. Yeah.
Wow.
I mean, maybe it was 6 weeks between the time they did the procedure and the time she played in that game.
Yeah, that's outstanding. Wow.
Yeah. And it was also really cool is that she was so inspired by the experience and just the growing up with the big heart zipper and other kids asking her about it all the time and the fear and being different and all those types of things. She actually wrote a children's book that just got published recently about her story and, her kind of going through, and that's sort of like her superpower.
Yeah, that's a pretty powerful testimonial. I think you may have your one of your next podcast speakers.
Yeah, I'm hoping to at least get her on a LinkedIn Live sometime to share that story because it was very inspiring.
That'd be really cool. Yeah.
Yeah. All right. I think now's a good time for new news. New news. So in Fierce Biotech on May 8th by Annalie Armstrong, in an article entitled "The Little Protein Factory That Could, Regeneron Gene Therapy Restores Hearing in 2 Children." So they developed a gene therapy to help kids with hearing loss caused by mutations in the otoferlin gene. This is similar to what we talked about actually a few months ago in January. Eli Lilly did this using a similar viral vector approach to restore hearing in 11-year-old. This study with Regeneron's in phase 1/2 called CORD is the name of the study. The 2 case studies that were recently presented at the American Society of Gene and Cell Therapy. In the study, the patients received is a little bit different technique. So they had the cochlear implant in one ear, and then they had the cell gene therapy in the other ear. So patient 1's an 8-month-old baby.
They served as their own control. Interesting. Okay.
Yeah, isn't that something? And so, so the baby began responding to sound after about 3 weeks of treatment and could hear even when the cochlear implant was turned off and continued to make progress since then. The second case study was a 4-year-old patient. Similar showed improvement after the few weeks, being able to respond a little bit, more verbally about what she was hearing. Mentioned that the cochlear implant had more of a screechy sound when she was listening to her parents talk, as opposed to the cell gene therapy, which is naturally fixing the gene, allowing the reception to get to the brain. But what made me kind of chuckle about this study a little bit was that the leaders of this research touted just how novel this study was when they and the cochlear implant does make it novel, but really Eli Lilly having a viral vector procedure had the same kind of result. So I thought that was kind of funny to me. So Andy, what do you think? What is your reaction to this? What do you think about the promises of these exciting cell gene therapies to maybe cure certain diseases and conditions down the road?
Yeah, absolutely. It kind of, my reaction also touches on a lot about what we have already mentioned, which is just the how crazy scientific advancement has been, and this is just another good example of that. I'm also kind of fascinated by kind of the child serving as their own kind of within study control, which I think is a bit innovative. But cell and gene therapy, again, definitely have been areas of focus. I'm a believer. Again, I probably go back to my earlier comment at being a bit therapeutically agnostic. Whatever the best solution is to the disease and the individual that's suffering from that disease, I'm all in favor of. I think we still have with cell and gene therapy. I mean, it is certainly more of the kind of trailblazing stage, which I think would be exciting, but that also means that we have a lot to learn, both from a positive standpoint, the positive pharmacologic potential, but also from a safety and tolerability standpoint as well. I think we need to be cautious, not skeptical, but just cautious about what the chronic risk potential might be for those classes of compounds. But some of the best stories that I had been part of, even really involved even cell and gene therapy and really antisense oligonucleotide specific with like the Melicin example, where we being the Boston Children's Hospital, Ionis, and Charles River, together with regulators, really developed an antisense oligonucleotide on the gene therapy that was so specific to the disease of this 6 or 7-year-old with Batten's disease that the product was named Melicin. So that degree of customization, which never, and again, 15, 20 years ago, we would have never thought about developing a tailored product for a patient population of one. That door would have closed for a number of different reasons, but I think cell and gene therapy opens the door to a whole different category of opportunities. So again, that just remind me of the Melicin example. So yeah, not hard to get excited by that, and also just recognizing myself how that example just ties in together with so much of what we've already chatted about. So most happy for the patient anytime we can make a situation better for someone in need. That's a success story. So.
And I can't agree more. And I don't know, Andy, if we all have, 10, 15 or 20 years left in this industry, but I think on our way out, we're going to see these really complex gene therapies are going to be, I don't know if routine, but normalized, right? It's not going to be every single one of them isn't going to be an amazing news article, which I think is great news in itself. When each one of these is just another drug, just another drug, right? And they're affordable because we've worked out those processes, and that's when we know that we really have made it in that space, I think.
Yeah, agree.
Absolutely. So, Andy, you're a past president of AAPS, the American Association of Pharmaceutical Scientists. And how has AAPS and maybe other similar conferences influenced your career and perhaps the industry as a whole?
Oh boy. Yeah, that's a big question. So I think I have been a member of AAPS for also over 25 years. I think the first meeting I went to was as a graduate student, and I feel like that was 1997 or 1998. I should know. I'm pretty sure it was 1998. And then that led to, again, a couple of visits to very early on, just seeking an opportunity. Like I enjoyed so much of what I experienced there as a student that it was just kind of easy to ask like the next question of how do I get involved more. And then when you have someone in your network like Dr. Ron Bauscher, who is just kind of an extraordinary mentor, and is, even then 20 some years ago, and as he is now looking for opportunities to return to kind of give back to people and to the community, that was easy for him to just plug me into some volunteer leadership opportunities at AAPS that took me from like abstract screening to chairing the biotech sections back when we had the sections and focus groups, to serving as member at large, to really being part of the group that started and unfortunately stopped, and then brought back the National Biotech Conference, which is going on now, and 100 different other ways that I was blessed to serve AAPS, and ultimately culminating in serving as its president, and during a COVID year, nonetheless. So getting to learn how to pivot from a face-to-face meeting to a virtual meeting, and then virtual meeting, and then a hybrid meeting. So all those things, I mean, AAPS is just kind of part of my DNA. And what I think about the most when I think about AAPS, and I hope this doesn't come off the wrong way, is I enjoy it mostly just because of the fellowship. I mean, some of my dearest friends and members of my network, folks that that I would sacrifice dearly for, and I know that they would sacrifice dearly for me. So not just professional friends, but true friends that you've established in different zip codes, different states, even sometimes different countries, and you not only stay connected through the year, but then you really connect each year at the at meetings like the National Biotech or certainly the FarmSci 360. So I have I've really enjoyed the fellowship and the networking. That's not to say I don't enjoy the science, because I really enjoy that as well. And it's the one association which brings a number of different scientific disciplines together, much like what we experience at work. And I still think that's a sauce it's a critical recipe. I mean, you've got some ADME, DMPK, bioanalysis, ClinPharm, PKPD, even a little bit of pharmacology, safety and tolerability, CMC. I mean, you're kind of bringing that, all of those functions that you would see representing a pharmaceutical company together at one meeting. Now, we've seen the industry kind of go through, again, sometimes folks just like their niche area, but I still like the value of bringing this community of pharmaceutical science together for not only the big meetings, but for all year learning content. When I had an opportunity to serve as its president, it was very easy. And my primary motivation was, why would I not want to give back to an organization for, 3 years? I think, your president-elect, president, past president, since it had given to me for, 25 years, that was just easy. And I continue to just try to be involved in AAPS as much as possible, because I think it is a great organization of professional peers and friends that I think makes just doing this journey of life, and even this kind of sub journey of pharmaceutical industry, pretty bloody enjoyable. So, and I would advise, young people that aren't involved in that kind of a community, I guess this would be all of our heartfelt, get involved, because you really do, life, it was never intended to be done alone, but really in partnership with others and fellowship with others. That's really will ultimately, I think, help make you stronger as an individual. So.
And Andy, you were leading really at a critical time with AAPS in a few ways. And one of the things, though, for me, when I joined in the late 90s, I remember someone telling me, you should join because you're going to go to the meeting anyway, so you'll get a discount, right? And then I'd go to the meeting once a year, and I didn't really understand the connection that you would build. But I think where that transition in the last 10 years, the organization has become an organization and a society of the people, like as you discussed, but then we also have great meetings, right? So we're connecting throughout the year with all these different activities, and we connect even outside of AAPS with, maybe it's emails, maybe it's phone calls to get advice through LinkedIn. But then we have these great meetings that are also part of it. But there's so much, and I know within the bioanalytical community, which I'm past chair there, we've done so much and worked so hard to bring people throughout the whole year. So it's just like, I feel like if you're in the US or anywhere in the world, really, and you're in bioanalytical, you gotta be involved in the bioanalytical community at APS if you want to really stay current and stay up to date on what's going on. It's become a really powerful tool for me to learn and keep up with the industry. So absolutely, I agree. And and I'm personally thankful for all your volunteer leadership for AAPS and others. And I know Greg knows the association, the professional worlds very well also. So
indeed. No, I love going to these type of conferences, and had, like I said, a great experience myself at MedCon a couple of weeks ago. It was really set up so well for networking and a lot of great information. So totally worth it. Yeah. Chad, do you have any other things that you'd like to cover?
I don't. Boy, we covered so much. Like, like every time we have these discussions, we get into amazing topics. I learn stuff about people that I've known for many years. Andy, I learned so much about you, not just facts and figures and reptiles and whatnot, but also learning about your philosophy, how you go about doing business, how you approach things, what really, what makes, what drives you. And I can't wait to see it at AAPS probably this fall, right?
Yes, I intend to be certain. I think that's Salt Lake City, if I'm not mistaken.
It is.
The last time we were at Salt Lake City was almost 20 years ago, if I'm not mistaken. And I organized a little half-day outing on the Provost River fly fishing. So I'm thinking about a repeat of that. So heads up on that, Chad. In case
I'm in. Yeah. Would love it.
We'll do it. We'll schedule that before or after the meeting, of course, so that we don't miss any content. So
for sure, love it.
Awesome, Andy. What is the best way for people to follow you or maybe get in touch with you?
Ooh, that's a good question. I'm not on much social media, but I am very present on LinkedIn. So you can certainly connect with me on LinkedIn. I have no issue. Even on LinkedIn, I think if you dive deep enough into my about information, you'll see an email. You'll even see a cell phone. So I'm not super private in that regard. And again, I'm always just looking for opportunities to bless others and just help provide some advice. Or if you have any follow-up questions, feel free to connect with me on LinkedIn. Send me a message, and looking forward to getting to know everyone that might do that, and certainly just been a privilege to join you guys today and share a little bit about me as a scientist, but even maybe me as a person and what motivates me in this in this journey that we're all on. So
absolutely. Well, I'll put your LinkedIn link as well as mine and Chad's in the comments down below. And Andy, thanks so much for just taking time out of your busy schedule to join us. It's been a lot of fun, a lot of great, interesting information. Like I said, we learned so much about you and about your approach and just more about the industry itself. Thank you, Chad, for all your help and support on this journey.
As always.
And thank you to all our listeners. We now have listeners, not just across the U.S., but across the world now. We have listeners across Europe, Taiwan, India, Japan, and Australia, which is really great. So thank you so much. Please do subscribe, share, like, comments. Let us know what you'd like us to talk about sometime in the future. And we do look at those comments. And in the meantime, take care, everybody. Thank you.
Absolutely.
Bye.